I have to admit, Charlie, I haven't prepared a wonderful description about you, but I can say from what I do know personally, Charlie is an outstanding researcher, has worked with Cortiva for a long time, is now into the university system, and he's very, very interested in this G by E by M and in the modelling and other aspects relating to that. So with no further ado, I'll hand it over to you, Charlie, and thank you very much for coming.
Well, thank you very much for the opportunity to talk today, but also for the opportunity to learn. I'm here because of my trajectory in the last about 20 years working with Mark and Graham. So it's not just by chance. I think there was a UQ opportunity training, and I'm probably an example of somewhat successful. I'm here. So thank you very much for that. Today, I kind of talk a little bit about the story I've been thinking about this breeding for climate change for quite a while, and we discussed quite a bit with Mark about this work. So let's start with where we are at, and often we get stuck in kind of this linear thinking. It's a good paper about the sensitivity of drought, whether it's relative, but essentially if the sensitivity of maize to drought increase and the future is going to be drier, there's a very straightforward argument to say we need to start breeding for drought tolerance pretty much everywhere, and that's kind of a sound argument. But then in PNAS we read that genetics has nothing to do with this, so maybe we need to stop breeding altogether. So, and these are pretty reputable journals, so where we stand is, to me, is to breed or not to breed, particularly for climate change, adaptation for crops. So key questions for me as I think about this presentation and the larger project is how this trait and system complexity underpin the shape of the solution space as we think about breeding for climate change, and importantly is, should we expect things that unexpected solutions, counterintuitive, the systems, they behave in ways that they are not straightforward as linear as we tend to think, and inform policy and investment and programs. The second question is, should we do anything about it, or the current systems that we have in place, whether the Corteva, Syngenta's and the like, can adapt as they keep working. If we let these quantitative genetic genomic selection phenotyping machineries move along with the changes in climate, are we going to move that germplasm at the pace that we will adapt to climate change and just do nothing? That's fine. It's been doing for a while so now. Where is the evidence? Do we have evidence to say yes or no on that, to answer that question? And finally, is about how we envision a crop improvement framework that is not just about the genetic gain, but some of that on top of the gluten-free and the oranges and nutrition, because calories, we solved that problem a while ago in World War II, but now it's more about health. It's about how you're going to reduce preventable diseases. $1 trillion problem in the United States, about $10 trillion worldwide, which when you think about the grants that we are trying to solve, the return on investment is absolutely ridiculous, but the problem is there nonetheless. And also how we are going to increase the system resilience and environmental health. And with that I mean, we cannot solve the climate change problem if we keep dumping CO2. So we need to first revert that problem, so our breeding objectives may need to be different than what we have today. So with those three questions, I will go through a couple of stories, and hopefully I will give you an insight of what I'm thinking and what is in my head for now. This is a project working with Ed Buckler, funded by FAR. So one thing is thinking about maize and the drought tolerance, and the other one is when you step out and you start looking at the whole food chain and what are the possible interventions as you think about synchronizing that supply demand of nitrogen with the intent of reducing greenhouse gas emissions, right? if you're going to make agriculture carbon neutral, the first thing you need to do is start stopping emitting greenhouse gases. So when you think about this circular type of economy framework, some things become not obvious. If you want to increase or improve the functioning of this system for warming climates, maybe cold tolerance is part of the solution to that, and I will elaborate that in a minute. So that's what I call the idea, the crazy idea person plus somebody has some credibility to the conjecture here. It's about breeding for cold tolerance to improve crop adaptation to a warming climate. And the idea here is if we can grow corn early in the growing season, we can have a much better supply and demand for nitrogen, and therefore we change the whole way we do fertilizations. So with that, we need to think about, well, what is the root of these problems, how cold affects corn, and I hope everybody agrees that this is a very droughty corn here, we're just in the field. This is a Mark's breeding program plot. The issue with that drought is when you look at the soil water content underneath, so above it looks drought, but underneath there's an ocean of water there. If you look at that sort of 7/22, which is the day we were on the in the field, there is water. The problem is why in the world that crop was droughty as hell. And part of the story, or at least the hypothesis that we thought is maybe is occupancy of roots. The roots are not growing, or maybe those membranes start getting into the mechanism, right? The membrane permeability is not functioning, so therefore the resistance is high. So with that idea, we start thinking, well, how deep are the roots in terms of occupancy? And the answer depends on where you are. In Canada, it's about 60 centimeters, and in Woodland, California, it's about 5 times of that. So essentially what this starts suggesting is there is sort of this limitation to root growth, which is invisible, essentially this is temperature. And among other things that I got ahead of me for a moment, that change with latitude, many things change with latitude, but one for sure is temperature. So with that, we hired Randy Clark, working with Mark in the group, and we start building root phenotyping capabilities to get into the mechanisms. That's what Christine mentioned a minute ago, to get our head around. Is there any merit on breeding for cold tolerance to improve adaptation to drought? So the first thing we wanted to do is about the occupancy. And after making some mistakes of, yes, the root doesn't grow, after we put this on a fridge is because the plant is not growing. So, okay, we need to now separate the temperature from the leaves, so photosynthesis is not compromised from the roots. And fine, but then the roots are not growing, or because the plant is not growing, because the membranes are not fully functional, and the nutrients are not going through the membranes, right? So we need to put some rings. You can see the pipes in there with the aeroponic solution. So the plant is well nourished. The temperature is not limiting from the aerial part. Of course, there's water because an aeroponic system. And then we just let the water table essentially in that sort of tanks fluctuate to keep the meristems cold. And we can do that for a number of different temperatures. And of course, we did the imaging analysis, so we can, as we cool these root systems, we can start estimating the rate of change for that root growth. Again, trying to get to the mechanism, to the root of the problem. And what we found is, yeah, as to start cooling the roots, about 10 degrees, which is 1.5 meters in Iowa, you have this limitation. It's like a rock bed. The plants cannot capture more water, and the reason why they are having drought stress is because they cannot get beyond that sort of depth. Can we do something about it? And this is where we start thinking about now building prediction. Is the genomic selection or identifying QTLs or genes that we can do interventions to break that sort of invisible barrier. So we went and screened about 200 inbreds. This is a diversity panel that you can collect from Cornell. And you can see that there are a couple of ways you can get roots deeper. One is by increasing the root on average, if you do the other one, and also changing that minimum temperature about 10 degrees. So there's genetic variation, therefore there's a solution to the problem. The other piece is, well, now we can get the roots down, is what these roots functional. So the question is, again, we need to get these root systems at different temperatures, we need to be able to pressurize so we can understand the hydraulic conductivity in those root systems. And of course, the aerial part need to be at non-limiting temperature. So that's a little bit of the rig that we have there. Hopefully you will see the pressure chamber that took a while to get approved by engineering, and now we can sort of pressurize the root systems at constant temperatures. So the fascinating thing here is, as if you build these light response curves trying to get to the underpinning mechanism is, yeah, you cool the roots and your photosynthesis goes down. So not surprising, there's no water. Essentially, the transpiration goes down, you cool the roots at 10 degrees, and therefore the growth goes down. So now you can start getting into the root of the problem. The other second question is, do we have genetic variation to solve this problem? And here what we can do is, as you start changing the pressure in that sort of chamber at the pressure at 2 different temperatures, what you can see there is that to maintain about 3 milligrams of water per square meter per second, essentially you need to almost double the pressure to get that water through those membranes, which are nice, they're not nice and fluid. So essentially, the plants need to do way more work to get that transpiration flow that otherwise is not going to move. We did a terrible experiment. For some reason, the technician thought 14 degrees and 0.5 was a good idea. That's we have a very little resolution of power, but it was enough to show that there is genetic variation for that hydraulic conductivity response to temperature. So again, now we have a couple of mechanisms of growing the roots in cold soils, and we have the ability to, or find genetic variation to get that water moving through the root system. So now that we understand the mechanism, essentially what the hell this means, okay? This is where we go back to models, and this do some exercise with models of different complexity. So the first question is, can we do this breeding by agronomy approaches to improvement to adapt agriculture to climate change? So this is a simulation. What if we plant corn in October? It's pretty damn cold in Iowa or in the Midwest in general. That corn survives and is ready to sort of start expanding leaves very quickly as the temperatures start rising. So instead of waiting for a couple of months until the plant decided it's time to grow, the plant is in the soil for months, but it survives at low temperature. What you can see is that if we can do that, sort of adaptation to cold, the water can flow through the root, there's a much better synchronism between the supply of light, the supply of nutrients, and you can increase 20% so the yield by planting earlier and making the corn cold tolerant is going to perform better under different environments, certainly under lower temperatures as you plant earlier. This is a work that it took me a while to get back, done by Graham. Was about explaining the past, right? Changing root angles. It didn't explain the past, but can certainly provide very strong insights about the future. It was about 20 years ahead of its time, because one thing that it happens in maize as you cool the root systems, the root angle just decreases, become more steep. And you can see in the simulation done by Graham 20 years ago or so, that if you change the root angle, we can increase yields, we can put more plant per square meter, and we capture more water in a way. So that's another piece of evidence from theory that yes, if we can increase adaptation to cold, in this case through the root angle, we can increase yields. And finally, we can integrate this with root system architecture. Hopefully you can see in the map blue point. That means when we make those roots grow faster, go deeper, kind of consistent with Graham's study, you can see year by year, 2012 was a drought, there are more blue dots, there are higher yields, so yes, we can increase yields when we make those roots grow in depth. That shouldn't be too surprising. What is surprising is that if we make those roots and membranes more permeable or to drought, doesn't happen, nothing happens. So what the hell is going on here? So let's stack these two traits and voila, there you go. In the drought conditions, if you get the roots down enough, it's an extra water. That water is not going to go very quickly through the root system unless those membranes are permeable. So essentially you start looking at some synergisms here that unless you have the two traits, not a lot is going to happen. Right, so some theory tell us that we need to stack traits. So the other things we can do with crop models, it's not only thinking about roots, but also how we are going to change the demand, how we're going to change the resilience of the reproductive structures, the hydraulic of silk elongation, and this is what you have in that slide. So the different colors in that graph, there are different traits, and the size of the sort of the areas under those curves is the importance of that particular trait to explain variation in yield when you run a multi-environment trial. So if you have different hybrids with different trait combinations, how important is, let's say, the root from velocity or response to temperature to yield variation at, let's say, 1,000 grams per square meter, pretty much on the middle. And it's pretty important. But if you push it to very high-yielding environments, there's water, so it doesn't matter that much whether these having more roots. And if there's no water, which is in the other end of, let's take to the extreme zero yield, there's no water, so root that doesn't matter. So these are non-linear responses. And as you think about the used corn belt, one thing you can observe is an increase in the area that is gray. At least that's what my eyes is telling me. And those is what is not explained by single trait. Essentially, 50% of the variation, at least, is explained by interaction between different traits. So even if you think about additive genetic models controlling individual traits, you're going to have 50% of that variation that depends on how these traits interact with each other. So now they're thinking complexity matters. It's not that simple as the single trait. It's that single trait in context of other traits and in context of the environment. And this is a good way to start expressing and understanding how climate change, moving this box left to right in different geographies, is going to change the important different mechanisms contributing to the adaptation. So I think that gets us to the complexity matters theory. I think we convince ourselves that, yes, if we can improve the response of these roots to cold, we can improve adaptation to warming and climate change by breeding by cold tolerance, at least in the context of some traits. So what the evidence is telling us. So that very nice is theory. The evidence is pretty scarce. Let's put it in those words. There's only one experiment that I'm aware, and Mark may help me honest here, which is conducted by Don Dubix starting 1970s, 52, okay. Asking the question, is these maize breeding programs contributing to increased genetic gain? So here you have pretty much every single experiment conducted at least with enough quality data. It covers a wide range of environments. You think Iowa is all rainy. No, in West it can be quite dry, and the East we have some nitrogen limitations. But bottom line is we can have now a data set that we can ask the question as the breeding program evolved over 100 years, how selection contribute to increase or maintain genetic gain while the environment was changing, while the management was changing. In these 100 years, increased irrigation, fertilization, planting dates, plant populations, you name it. So this is a snapshot of that experiment over that sort of 100 years of breeding, starting early 1930s about, that sort of orange-ish area. You can see that the amplitude temperature changed about 1.5 degrees while or during the same period of breeding. So the breeding machinery was making selections as for the same period of time, the climate was changing. Essentially, you have an experiment of how breeding responds to climate change, how breeding machinery can adapt to climate change. So the dots are hybrids that were selected by the growers. This is what the growers like. These are the successful. They know how to make money out of these hybrids. So they are robust, they are resilient, and they are what they call it successful. So between 1930s to 1990s, we have about 60 years of breeding, again, represented by the commercialization of these hybrids. They are non-transgenic, that's the main reason why we keep it, so we have a confounding effect removed. And we can think about, okay, what if we run all these hybrids, or at least most of them, in the next 30 years while the climate was still changing? So we have environment changing and genetics changing, as you can see from that graph, over about 60 years and 30 years of experimentation. That's what these era hybrids enable us to do. So in this matrix is a different way of representing the same what I said. In the rows you have genotypes commercialized between 1980 and 1991. So you have all these rows, the G component. In the columns you have each year, there are 30 years where we repeat the exact experiment with the same hybrid, and we did it on the 3 different management conditions. And my we is that I conducted about 5 years of these experiments out of the 60 or 100, I don't know how many. So my little wee contribution. So when you have this for each year, now we can do the Dubix plots, so we can calculate the rate of genetic gain. We just, on the x-axis you have year, but this is when the hybrid was sold or commercialized. Y is yield, the slope is the rate of genetic gain. And we can do the same thing for the, well, these are 2 years, but the three plant populations. So now we can plot the rate of genetic gain, essentially calculated, and ask a question, how it changed from year to year if we want to estimate that rate of genetic gain. So if you do it in probably close to 1920, it's close to 0, and the low plant populations, which is the figure on the left, some years you may estimate a ridiculous amount of about 7.5, but on average, when you have these isolated plants, you can think about this pure G potential. It's about 2.5 grams per square meter per year. So if you increase the plant population, because we don't grow plants, we grow crops, so managements matter. This is a population trait, not a plant trait. So when you plant this at the right plant population, you can multiply that rate of genetic gain about 3 times. It's a little bit of a trend, but let's assume that there's no trend there, which is, and I think it was surprising as we analyzed this data with Mark, is when you push these hybrids even higher, and that's, again, the climate was changing in the last 30 years, the rates of genetic gain were increasing, not decreasing. That's what's the intuition of what other scientists are advocating, is as a climate change, we need to start breeding for climate change, right? It's drought tolerance, blah, blah, blah. So what the evidence is telling us is that no, the breeding machinery essentially is enough to keep track of the changes in climate. We can go through that theory more in detail, but essentially all the rate of genetic gain, if you don't even believe on the acceleration, they are positive in spite of the changes in climate and management. So the answer to do we need to breed for climate change, which was my second question in the talk, is the answer is no. It's just fine. We don't need to do anything. There's a caveat, right? The caveat is if we are going to use the same amount of fertilizers and we are going to continue greenhouse gas emissions and using ridiculous amount of water. So the problem of breeding for climate change is not whether the breeding machinery can increase yield and adapt to the changing climate. It's more about changing breeding objectives. So agriculture is part of the solution and not the problem of contamination. So that is the message here. Is we need a different framework to do breeding to solve the problems that agriculture is changing, not because that machinery can deliver on the needs of society. That's for maize. We'll talk about biodiversity. That's a very well point, Christine. You say 4 crops, this is one and a major one. But look at what happens with sorghum, at least in the United States. This is a paper we just published in 1960s. We have plenty of areas, to grow corn. Sorghum nowadays is a species in view of extinction. The answer is simple. We don't invest in corn. The rate of genetic gain is low because we are not testing enough germplasm. Okay, fair enough. You can see there different evapotranspiration levels. The rate of genetic gain in sorghum is pretty much flat everywhere, and the corn is positive. The Alcomax accelerate that rate of genetic gain. Is that simple? Maybe, maybe not. So to understand that, one thing we can use is this breeding gap analysis, so this cloud of the possible framework that Mark developed when we were in Corteva. So we can run this combination of genotype by environment by management combinations and plot it against a resource that matters, in this case evapotranspiration and yield. And within that cloud, we can start understanding what are the genetic combinations and management that can give us to where we are today with the germplasm to where we need to be. So within this heat map, one thing we can do is do the same thing of cloud of the possible for sorghum. Can this methodology can help us understand what's going on with the rate of genetic gain? So that's a cloud, is certainly different. We run about 700 million simulations. Doesn't take a lot, but still quite some work. And these clouds start helping understanding quite obvious differences that we know that exist in sorghum and maize. For example, yield potentials in corn is about 20 tons. In sorghum, at best, in a good day, is going to be 14. The other one is between 400 millimeters. Maize, it has a million simulations with 0 yields, and you can see a smear of red there. It's telling us there's a risky proposition there. When my student told me he couldn't do that, I told, well, you should think harder, go learn how to program. He said, no, sir, there's 0 in the data set. It's not doable because there's no simulation with 0 yield in sorghum. So now we can start understanding what's going on between these two crops. So now we have a framework, we have this cloud of the possible. So now we can overlay the data from Rotundo and Borras and company, and I'm the last one there, a little bit refusing because I wasn't quite sure what's going on, onto the cloud of the possible. And there's a battery running low, and I'm going to have to talk a little bit different here. So think about, better start moving, entertain you a little bit more. Each of these points is one hybrid commercialized at some point in time in the last 60 years, or more than 80 years, from 1960s. So the first thing you observed is, unlike corn, the hybrids are pretty much bumping against that 99 percentile. Essentially, sorghum, when you grow in the absence of abiotic stresses and in a range of evapotranspiration, they are reaching that biophysical potential. So if hybrids are reaching a biophysical potential, would you expect to have any genetic gain? No, they're there. For the context of the set of biological or biophysical traits that characterize this germplasm, this breeding program essentially is stuck in a local optima in the adaptation landscape. That's why they are not making any progress. It doesn't matter which is the level of evapotranspiration, they're just stuck. They're just chasing the tails, looking for combinations, and cannot get it out of there. The interesting thing is you see the dotted line in the upper plane that fits that sort of a quantile. It's a dotted line because we didn't simulate 900 millimeters of water. Those are the things you can do in Viluco in Chile, but it's not something you're going to simulate for the Midwest. And when you extrapolate the quantile we estimated, we pretty much nail on the observations in Chile. So it tells us that the framework is quite functional. And it doesn't matter what level of evapotranspiration, there are hybrids that are hitting that boundary. So it's not investment. We can put more money, we can do more crosses, but if there are hybrids that reach that boundary, we are not going to cross it. Doesn't matter what recombination we are going to get. We already found them, in other words. See what's happening with the breeding program, at least for drought tolerance and yield potential. In 2008, it was reached the peak of drought tolerance in sorghum, at least in this germplasm. And in 2020, pretty much hit that boundary on the high evapotranspiration. What I would like you to note is, where is that yellow dot on low evapotranspiration? So essentially what is happening here is they're generating crossover interaction. There's a lack of a correlation now. Not only they're chasing their tails, but they're pushing yields higher. They're trying to grow it in a better environment, so they're losing the drought tolerance in sorghum in order to increase yields. So essentially that sort of resiliency and productivity is being compromised, chasing or trying to find solutions around this local optima. So now we can ask the question, all right, how are we going to get out of there? What are the possible combinations or trait combinations that we can explore or create, whether it's using gene editing or branching or tillering, you name it, again, whatever the technology, in order to get out the germplasm out of that local maximum. And essentially what we did is run a simulation, do the multi-environment trials the same way as explained before, and quantified for these 5 traits what is the yield variation explained by each of the traits. So under relatively high yielding conditions, more of all the traits contribute in a relatively predictable manner. High water availability, high radiation efficiency, check. What I would like for you to note is in maize, it was about 50% in that target population of environment, which it was about 10,000, gram per square meter, sorry, it's 10 tons. But when you start getting into these lower environments, look at the arrow in there, which is the target population of environments, 250 to 400 millimeters of water, is 80% of the phenotypic variation results from the interactions of these traits. Not only these breeding programs are stuck on a local optima, it's impossible to find the right combinations because they are going to be chasing one thing. You squeeze the germplasm here, you select for one thing, and it's pop up in somewhere else. It has a compensation, if you will. So it's very difficult to do breeding when you have this, even if the additive genetics is going to be not additive physiology. And that's what is shown there. So it's very difficult it's not only less investment, but more difficult to find, and it's stuck on a local optima. Anyways, I think that's a different story that for sorghum is not that straightforward as in maize, or at least this evidence suggests that maybe adaptation to climate change is going to be a little bit different than for maize. So now going back to the final point I would like to talk about is, can we envision a framework to breed for climate change? And the first thing is, it has to be multidimensional now. The first, if you will, arrow in there, in the diagram is about multiple traits, is the figure I show you. That scan across yields or evapotranspiration and trait combinations or gene combinations. So fine, it's just one. But with simulation, the other piece we can do is we can start quantifying what are the greenhouse gas emissions. These are not be able to phenotype those things. What is the water use? What is the water use efficiency? Traits that are much more difficult to evaluate in the field conditions, and not to mention carbon sequestration that happens in a very long 10s to 100 years time frame. So again, this is a bit of the crop growth model help us to enable this framework. And this is what kind of my cartoon diagram of a crop growth model. Graham would be laugh at me, but what I really want you to pay attention that even with that simple crop growth model, which are 2 equations, just follow the W's in that two equations diagram. You see W in the right-hand side and the left-hand side. You have W in the 2 equations. You have theta, again, on the right, on the left, and the 2 equations. What happened with these type of systems of differential equations? They oscillate. So they're very difficult to predict. They have multiple optima. And it doesn't take a lot to write a model of a plant, it's right there, to generate complex patterns. And not to make things complicated, but now to bring some formalism on how we can bring together the genomic selection with the physiological framework, so now we can look at, let's say, theta or omega, or maybe all the other parameters, K1, K2, K3. We can model any of those as a function using the simplest thing, which could be the Henderson animal model or any gene to phenotype or genomic selection algorithm of your choice. But here is transparent how we can link information, whether it's markers mainly, at least in my head, to traits, and let the crop growth model do the interactions to tell us where in these adaptation landscapes it may reside, and how we are going to move populations from one place to another. Because these are complex systems, at least as you think in those two equations, it's not as easy as do an inverse calculation using optimization. You start using some Bayesian approaches to implement what I will call abductive reasoning. So essentially, we need to have a family of hypotheses that we need to test. And of course, it's going to take phenomics and all the omics in order to generate the data to be able to train this system. So getting close to the end now we have the tools, we have how to connect map gene to phenotypes, and we know at least in sorghum that because we are trying to force and we are stuck in local optima, we start generating with the crossover interactions or lack of correlation, which essentially is going to generate a graph that may look like that. And here we talk about selection environment and evaluation environment. This could be the low evapotranspiration and high evapotranspiration. So imagine if you're selecting corn, which is one of the, look at on the X and Y or the plane on the base. Let's say P in selection environment is a fraction of low evapotranspiration, and we are going to predict on the high evapotranspiration. Or if you are in the 2020, we are going to select more for high yield and high evapotranspiration, how it's going to perform in low evapotranspiration. Meaning they're going to be a crossover interaction, right? So depending on the frequency of environments that are included in that multi-environment trial, you are going to have different responses and shapes of this graph. And you're going to see a correlation between P and F can be present and future, or it could be selection and high or low evapotranspiration. Sorry, I got a little bit stuck on the on the abbreviations here. So to recap, if you are percent P in selection environment, there is 100% drought stress, and you want to predict and the well water conditions, I show you in the corn example, that's a bad idea because it's going to revert the rankings. So your WG, that genetic correlation, is going to be low or even negative. Well, it's going to be negative. The opposite is going to be true. What are the implications of this result when you have this lack of correlation and your correlation between now thinking about well water condition of today and drought into the future, P and F, is if you are selecting for today's environment, you're going to select, let's say, in well water environment for well water environment. So that's good. You're going to fit current populations nicely. If you're going to do the opposite and say we are going to select for future population, let's say is drought, we are going to be fine. The problem is if we select for the future, because of the negative correlation, select on the well water, on the drought conditions and today we have well water, we are going to starve today's populations. And if we don't breed for the future, we are going to starve the future populations. So now we have a framework that is not about if we need to breed for climate change, is how we are going to do it, how we are going to time. It's about timing when you need to start the transition from today's environments to future environments, making things all way more complicated because you wouldn't want to spend all the resources in today and don't care about the future because you're going to starve the future populations. But we don't want to go too fast because you're going to starve the current one. So that timing is of essence here. A little bit about advertisement. I thought I will include this because part of the solution, it could be gene editing, certainly in fruit and vegetables. And that's what we are trying to do at University of Florida. what we like to do, and this is more for discussion than anything else, hopefully I will motivate the scientists in this group to think about. Unlike in other times at University of Florida, we started focusing on products, and the products, these come from the greening, the citrus greening in Florida is devastating the industry. So that forces us to rethink how to do biotechnology, if you will. So we start working background from product to program, if you will, from product to concept, but it's an iterative framework. Essentially, it's an end-to-end, if you will. So what we are building, or at least I'm trying to build, is a set of foundational capabilities going from prediction in AI that uses crop growth models as bioinformatics, if you will, to think about ideas, building plant transformation labs and speed breeding so we can generate and take those plants to the field as soon as possible. We have a close interaction with the breeding programs because traits and genes don't sell on a tube, they sell with germplasm. Unless they're in the right germplasm, they're not going to be adopted by farmers. So that's a way we have this sort of intersection between breeding programs and the center. The other thing is we are building regulated sites from North to South Florida, covering a fairly wide range of locations and robotic capabilities to do the phenotyping. And finally is the business development. We need to get this on the hands of growers, going through regulatory and some of the things you talked, Christine. So hopefully, as we think about what we have in Florida, what we're building in Florida, and what the center is looking to do it in Australia, I think there's an opportunity here to do research and start integrating and create critical mass on how we're going to take ideas and deliver it into products as fast as we can. Because as I said, time is of essence. We need to deliver or do breeding at the speed of need, essentially. So here, just a little bit of data on AI prediction. We have NVIDIA donation and support to implement models in supercomputer at IFAS, building, where we have a couple of faculty positions, building indoor agriculture facilities to grow the plants. We have about breeding programs for 50 crops, so you name it, pick the one of your choice. We are going to have gene editing capabilities to do that, about 7 sites to do regulated testing all throughout Florida, and of course, university regulatory licensing and extension. We have one person in each county in Florida that is going to help the growers use and find that genotype by management for the environments that they are experiencing in that part of the state. So I think somehow we converge, and I think there's a tremendous opportunity to work together more than ever before. So with that, thank you very much, and I will leave this with this cartoon that breeding for climate change is far from simple. It's not the investment, is the complexity of the trade by environment system. We need to think about it, we need to understand regulatory mechanism. Throwing money at it is not going to cut it, and we need to do system-level thinking. We need to have monitoring networks. We have only experiment, like the air experiment in maize. We sort of twisted some ideas on similar experiments in sorghum conducted in the US, but the rest of the crops we are navigating blind. We are thinking about breeding for climate change, and in the future, we have no idea where we are. And certainly we need to harness all sources of variation, so it's not genomic selection, it's genomic selection and breeding and gene editing, and depending on the complexity of the trait, we will be able to get germplasm out of these local pigs as we have in sorghum in the United States. With that, thank you very much, and thank you for getting me here after a 20-year journey. Thank you very much. Questions? That's good.
That's a great talk, Charlie. I was just looking at your last slide. I was a bit disturbed to see the AI at the start of your process, and kind of assuming that your crop growth model understands the mechanisms well enough to rely on a computer algorithm to drive it. Do you think we understand those mechanisms well enough, especially below ground?
Good question. I try to convince you that we have some idea of what's going on below ground on the temperature. Yes, the AI, think about the pillar is a capability, not necessarily we understand it. I think is Graham call it a roundabout, right? Yeah. So you model things and you're trying to understand it, you do experiments and you improve your model, and it's iterative, right? But for example, in the case of citrus greening, we have some understanding, but certainly it's incomplete. We don't have time. In 5 years, the industry is going to be gone. So the model can help you put hypotheses there that are testable. The sure thing is, if we don't do that, we have zero at the end of the pipeline, right? If nothing goes in, nothing is going to go out. So the best shot is use theory and the current understanding to put hypotheses and think about this pipeline as an experimentation pipeline. It's a learning pipeline. Because you're going to, after you put these things on as you put the transformations or gene edits in the field trials, they're going to tell you how they're working. So you take that knowledge and you feed back to the AI and you keep going. So it's not a product development here. It's an engine that is going to generate, if you will, possible products, but along the way is going to generate knowledge that everybody can use for publishing, understanding, testing. Again, what goes in has to have some resemblance or a chance that is going to help the grower at the end, right? It's curiosity-driven, but some application constraint, if you will. That's what Mark learned from the master here in the breeding program, right? You can do science that can lead to better cultivars.
Question up the back. Was there a question up the back? No?
Hi. Beautiful talk. So if I got it right, what you're saying is that you're predicting you're going to use gene editing to sort of turbocharge what you can get from the background of conventional breeding. And so you seem to have confidence that with such an investment in gene editing, that it's going to be a success. So my question is, what are the sorts of traits you think you're going to be editing to turbocharge for the future?
I can tell you a couple of, well, one is my favorite, work in broccoli, right? And broccoli can take the heat, no problem. But it's developmental, right? So the development is that messing all the branching, and what used to be a head and the cold environment become all sort of fluffy and disconnected networks on the high temperatures. So you can start thinking about development and looking at QTL information to start doing gene edits, so to make that responsive less. And we know there are cultivars from 100 years ago, from the Roman times, that they are nice and pretty. But they can take the heat. It's the modern germplasm that doesn't take it because it was selected for cold environments. So it diverged, so we need to now bring it back. So sources of gene edits, I think, is the knowledge that Christine is going to generate, to publish. For example, in citrus, right? One of the things we are doing is using AI to mine the literature. So essentially, we are generating AI hypotheses that is driven by what we can scan and understand the integrated body of the literature on citrus greening. So do we understand perfectly? Absolutely no. But if a human can sort of get the hypothesis, AI probably can do faster. So it's like a research tool? Absolutely. Well, product development is a research path, right? With the outcomes are more applied. Is the case study that kind of matters, or you classify as applied or less applied, right? Last week, I was in the Gordon conference, someone said, well, I'm very interested on how quick the stomata was fluctuating. So that's a curious-driven idea. I'm not sure I'm going to do any edit until I'm convinced there's a little bit more merit on how that sort of opening and closing is going to affect yield. We can use models to test that hypothesis. We can rank the merit. You put it through the pipeline. The empirical evidence will tell us whether you're right or wrong. Yeah, it's...
Cool. Thank you so much, Charlie.
Thank you.
Yeah, look, another round of applause for a great presentation. Thank you. Just another little health check. You'll be perhaps wanting to find where the toilets are, so they're up the back and to the left. And if we go outside here, it's out a little corridor near the lifts on the left-hand side. So now we're breaking for morning tea. And something else? Back at 11, yeah.
So morning tea's on the lower level here. If you just go out this section here, you'll get it. But please, back at 11, seated.